Shri Gurubhyo Namaha. I welcome you all to our online teaching academy Ekaliva. Today in this video we are going to start another important tool what we are using in our RDNA technology, that is our cloning vectors. Okay, in our last few videos we have seen about our restriction enzymes and our other enzymes what we are using in our RDNA technology, and today it is cloning vector, and you all know what is a cloning vector. Okay, so vector actually it is a substance which is carrying a substance from one place to another place, that is which it is acting as a mode of transport. For example, in Plasmodium infection you can see that Anopheles mosquito is acting as a vector to carry that disease causing organism from a diseased person to a healthy person. Okay, in the same way here something different but doing the same function. So cloning vector, they are the vectors which are going to carry a gene of interest from one source to the other. Okay, so this cloning vector, when you see this, okay, it is a small piece of DNA. Okay, a cloning vector, it is a small piece of DNA. Commonly it is present in few viruses and bacteria. Okay, bacteria, okay, it will be present in the form of plasmid. Okay, and these DNA fragments, okay, they can be stably maintained in another organism. Okay, it is isolated from one organism. When it is inserted into some other organism, it is so comfortable to maintain all its structural and functional forms. Okay, without any disturbance it can do its function. So that is actually the special thing about our cloning vectors. So most commonly used cloning vectors in RDNA technology are two, that is our plasmids and bacteriophage. Okay, these are the two main vectors which we are using. And when you see the salient features of cloning vector, okay, which is making them as an ideal vector, when you see the first one, ability to replicate within the bacterial cell. Okay, when this bacterial cell, it is host cell. Okay, when you insert this cloning vector or introduce this cloning vector into some other bacteria, they are so comfortable and they can replicate, okay, independent of that bacterial chromosome. Okay, they can replicate by themselves within that bacterial cell. Okay, that is the first feature, and these vectors are having a high copy number. So what is that copy number? When you see the alien DNA in the sense our gene of interest, okay, that is a foreign gene, okay, which we are going to use for production of some useful products. Okay, that foreign gene we will call it as alien DNA. Okay, when this alien DNA integrated with the vector, it multiply equal to the copy number of vector. For example, the plasmid copy number is 15. Okay, our gene of interest, it is inserted into the plasmid and that plasmid introduced into a host cell. Okay, so when its copy number is 15, within that host cell in one E. coli, it can produce 15 copies of that plasmid. So that is called copy number. So vectors that these vectors, okay, the cloning vectors, they are having the copy number, they are supporting the copy number from 15 to 100. In the sense, we can produce 15 to 100 copies of that plasmid in our host cell. Okay, so that the product amount can be increased. And these vectors can be engineered for easy linking of foreign DNA, and also, okay, they are structured in a particular way to select our desirable genes. Okay, we will see, okay, in our following sessions, okay, we will see about the selection and all. Now, we are going to see an important parts or features of our cloning vector. Okay, to be an ideal cloning vector, okay, it should possess all these four properties. Okay, origin of replication, okay, which you will indicate it as ORI. Okay, origin of replication, selectable markers, cloning sites which can be otherwise called recognition sites, and vectors for cloning genes in plants and animals. Okay, we will see one after the other. So the first one, okay, we are going to take one example, okay, one plasmid as an example, and we are going to see all these features. Okay, and the plasmid which we have selected is PBR322. That is the name of the plasmid. So what is the PBR322? When you see this P, okay, it stands for plasmid. Okay, it indicates it is a plasmid. And this B and R, it comes from the name of the two scientists, those who have prepared this PBR322. Actually, the plasmids, they are of two types. Okay, naturally occurring plasmid, which are naturally present in the bacterial cell. And this PBR322, it is actually okay, an artificial plasmid, okay, which has been prepared by combining the genes from different sources. Okay, so that is an artificial vector, and this artificial vector prepared by two scientists, Bolivar and Rodriguez. Okay, so from their names only, this B and R, okay, Bolivar and Rodriguez. Okay, and what is the 322? The number, when you see, it is identification number. So in the same laboratory, they will be identifying or preparing so many plasmid. Okay, and this PBR, that is the 322. Okay, it is to distinguish this vector from other vectors which have been developed in the same laboratory. So something like identification number. So this is our PBR322, and in this PBR322, we are going to see all the salient features of a plasmid. Okay. So the first one, origin of replication. Already I told you, and you all know what is the use of origin of replication, because already in our second unit, okay, you've learnt about that replication process. Okay, origin of replication. Okay, something started here. Here it is going to be our replication. Okay, so the sequence on DNA where replication starts. This is the first function of our origin of replication. Okay, that is the actual segment from where the replication is going to start. Okay, and it is having another important work. Okay, so the alien DNA, when it is linked to it, okay, it will replicate with the host, within the host cell. So at this origin, the replication starts. Okay, so you are going to insert your gene into this plasmid, so that gene also replicate along with its plasmid DNA. And what is the second function of this origin of replication, that particular segment of that plasmid? When you see, it regulates and controls copy number of a linked DNA, that is our foreign gene. Okay, so two main functions. The first and most important function of our origin of replication, it is to start replication. Okay, and also it controls the copy number. Okay, this origin of replication, it is responsible for the number of copies going to be produced. So when you are selecting the plasmid, okay, you have to look for the plasmid which supports high copy number. Okay, always you have to select a plasmid which is supporting high copy number. So this is about the first segment of our cloning vector, that is origin of replication. Okay, the next one, selectable markers. Okay, selectable markers, okay, they are going to use, okay, we are going to use it for selecting something. Okay, so this plasmid we are going to introduce into the host cell. Already you have seen the cloning process. Okay, you will be taking a foreign gene and the plasmid DNA, and it has to be restricted with the same restriction enzyme. Then both this foreign gene and the plasmid DNA, it is ligated together, okay, that is sealed together. Then after this, okay, we will call that particular combination of plasmid DNA and foreign gene as rDNA, recombinant DNA, and this recombinant DNA is introduced into the host cell. Okay, when you introduce them into the host cell, okay, it is not possible for all the host cell to take our rDNA. So some bacteria will have our rDNA and some other bacteria, they will not take that rDNA. So the bacteria which is having our rDNA, we will call them as our transformants, and the bacteria which are not ready to take our rDNA, which are not having our rDNA, we will call them as a non-transformants. So this selectable markers, okay, they are used to identify and eliminate non-transformants, because no use of non-transformants, because they are not going to have our rDNA. So, we will eliminate those non-transformants and we can select only the transformants, that is our bacteria with the required rDNA. So, we need to select only those bacteria, that is transformants. So, for this selection of transformants, okay, we are using a segment of our plasmid that is called selectable marker. Okay, and this selectable marker, when you see, okay, mostly they will be using a antibiotic resistant gene. Okay, the resistant gene which is okay, giving them the resistance to some antibiotics. Okay, antibiotics, you know, the substance which is killing the bacteria. Okay, and here what are all the markers, antibiotics used when you see ampicillin, chloramphenicol, tetracycline, kanamycin. These are few examples. Many more antibiotic resistant genes are there. These are few examples for antibiotic resistance gene. Okay. And so, I told you, selectable markers. So, when you take our plasmid, that is PBR322, it is having 2 antibiotic resistance gene, okay, segments, that is 2 selectable markers. So, you can see some pink color structure, here one pink color structure and here one pink color structure. Actually, that is our selectable marker, and this particular segment of this DNA, this particular gene, it is responsible for conferring resistance against the antibiotic called tetracycline. Okay. So, this is tetracycline resistant gene and this is ampicillin resistant gene. So, PBR322, it is having 2 selectable markers. Okay. Using this particular segment of DNA, we are going to select our transformants. That is the important use, okay, significance of these selectable markers in our cloning vector. So, we completed origin of replication and selectable markers. Okay. The next important part of a PBR322 or any other plasmid when you take, it is our cloning sites. Okay. What is a cloning sites? Cloning sites, otherwise they can be called as restriction sites or recognition sites. Okay. Restriction sites or recognition site. So, restriction site in the sense the site where our restriction enzyme is going to cut the DNA. Okay. And recognition sites where we are going to insert our foreign gene. Okay. So, this is our cloning site. So, the use of cloning site, it is to the site of the DNA where we are going to clone our, where we are going to insert our gene of interest. Okay. So, that is cloning site. Okay, actually this PBR322, it is having many cloning sites. I will show you. See here, these are the cloning sites present. See, PALM-H1, SAL1, PVU2, PST1, PVU1, ECHO-R1, CLA1 and HIN3. Okay. Something, okay, somewhere you have seen all these things. What is that? Actually our restriction enzyme names all this. Okay. So, this is the site when you use restriction enzyme, for example, PALM-H1, that restriction enzyme will go and cut the DNA at this site. So, it will cut open the DNA. So, the plasmid DNA will open like this. Yes, the plasmid DNA will cut open like this. So, if you have your foreign gene, okay, the foreign gene also treated with the same enzyme and that foreign gene can be successfully inserted here in this place. So, foreign DNA is nothing but our alien DNA, gene of interest. Okay. So, that is actual use of this restriction site, cloning site. So, PALM-H1 when you use, it will go and cut here. If you use SAL1, it will come and cut here. So, like that any enzyme, okay, whichever enzyme you are using, already you know each and every restriction enzyme, they are having their own restriction site. So, in that particular place they will go and cut. And one more important thing is, when you select a that is plasmid, any plasmid, the plasmid should have a single cloning site for an enzyme. Okay, for example, suppose palm H1, it is only one site. You can see all the restriction enzymes are having only one restriction site. Suppose, okay, this plasmid is having two restriction sites for this palm H1. Okay, here one more palm H1 restriction site is there. Okay, if it is so, what will happen? When you add restriction enzyme, that particular palm H1, it will go and cut DNA here, that is plasmid DNA here and also here. So the plasmid DNA will not open like this, it will cut into two different fragments, okay, which will make our cloning procedure very difficult. Okay, so you cannot clone your foreign gene like this. So because of that only, the plasmid DNA, that is any plasmid, okay, here even PBR322, when you see, it is having a single cloning site for all the enzymes, okay, that is restriction enzymes. So this is actual role of cloning sites in our plasmid, okay, in this PBR322, and something is given here, you know that. So EcoR1 already you know it is an enzyme, okay, which has been extracted from E. coli. So nomenclature you already know from the bacteria, from which organism they were isolated. The first letter stands for genus, the second two letters stands for species of that particular organism, and this is a strain name and this is a series in which it is prepared or isolated, okay. So and hint to you know Haemophilus influenza and palm H1 in the sense Bacillus amyloliquefaciens, and CLA1, okay, it is Cariphenol lalunum, and SAL1, Salmonella albus, Proteus vulgaris PVU, PS2 Providentia stewardii. So these are the bacteria from where they have isolated that particular restriction enzyme. So this is about our cloning sites. Cloning sites used to insert our foreign gene. And okay, and in addition to all this, we have seen about origin of replication, what is that, selectable markers, you have seen about all this cloning site, and you have a small segment here, okay, which is labeled as ROP, okay, that is a segment, a small segment of gene which is going to code for a protein, and that protein is very essential for the replication of this plasmid. So this ROP stands for replication of plasmid, that is a segment of gene which encodes a protein which is essential for the replication of plasmid, okay. So this is about the parts of our PBR322, okay. Like this, each and every cloning vector will have their own parts, that is common parts. When you see, they should have an origin of replications to start replication, they should have a selectable marker to select our transformants, and cloning site should be there to clone, to insert our foreign gene, and then ROP should be there, okay, to take care of the replication of plasmid. So this is about the features of our plasmid DNA, okay. So this plasmids and all, it is used to introduce our foreign gene into bacteria, okay. So the host is bacteria there. When you take PBR322, the host of that PBR322 is bacteria, okay. In the same way, okay, some plasmids, some cloning vectors are prepared to transfer genes into plant body as well as animal body, okay. So we are going to see two examples for that, okay, that is vectors for cloning genes in plants and animals, okay. There is a bacteria called Agrobacterium tumefaciens, okay. This bacteria, actually it is having a plasmid called TI plasmid, okay. It was having a plasmid called TI plasmid, okay. What does the TI stands when you see? Tumor inducing plasmid, okay. When this bacteria infect a plant, it introduces this bacteria and it will induce tumor, okay. That disease we used to call it as we are calling it as a crown gall disease, okay. So this TI plasmid, scientists focused on the feature of this Agrobacterium. Once it is infecting, it introduces this plasmid, and this plasmid is having a special segment called tDNA. This tDNA is actually responsible for producing the tumor. So that tDNA, it is introduced into the plant body and it is causing the disease. So scientists concentrate on the feature of this particular bacteria, and what they have done, they use this TI plasmid as a cloning vector to introduce a gene of interest to the plant body directly. Okay, so how? Okay, and you can ask me the question, when this TI plasmid introduced into the plant body, it can cause that crown gall disease. How they are making it? When you see here, when you insert, okay, actually in this place exactly they are going to insert our gene of interest. Okay, so when our gene of interest, it is introduced into that particular segment, the tDNA is getting inactivated. In the sense, it is losing that pathogenic activity. Okay, it is losing that ability to cause the disease, but still it can transfer the gene into the plant body. So, okay, it is not going to cause disease. At the same time, our gene can be transferred into the plant body. So, this is how scientists concentrated on this agrobacterium to use it as a plasmid, that is cloning vector, for introducing the genes into this plant body. So, the gene is directly introduced into the plant body so that whatever changes they want to do with that plant, they can make it. Okay. So, this is one important plasmid that is cloning vector, which is used to introduce genes into plant body. The next one, in the same way, there is a virus called retrovirus, and this retrovirus is also having the same ability to transfer the genes into the organism where it is causing that infection. Okay. So, these things, okay, what they will do, they will remove the particular gene which is responsible for causing, okay, that is pathogenic effect, or else in that particular area they will insert our gene of interest, so that this bacteria, when it is introduced into the animal cell, it is not producing the disease, instead it is transferring that foreign gene. So, two vectors, okay, PBR322, it is a vector for transferring genes into E. coli bacteria and Agrobacterium tumefaciens, and this retrovirus respectively, it is used to transfer genes into our plants and animal cells. To receive our online lessons, please press the subscribe button and you will receive the latest update. Thank you, children.